My name is Junhao Wen, and currently I'm an assistant professor and the director of imaging genetics at the Columbia Radiology, Computer Science, and Biomedical Engineering departments. And my research mainly focuses on using advanced computational methods like machine learning and AI, develop and apply those advanced methods to different kinds of biomedical data, including in vivo imaging data, like brain MRI, but also multi-omics data, like genetics, proteomics, and metabolomics. And the area of interest has always been aging and Alzheimer's disease for me, like neurodegeneration. So we have recently developed what we call biological aging clocks to digitize individualized risk towards like aging and Alzheimer's disease. I think I know this YouTube channel because I have followed Dr. Reitland's work regarding like sleep, and of course I think in the sleep domain, her work has been like fundamental and one of the very earlier pioneers. That's how I heard about this channel. It has been very critical for this work. In the paper, the main resources we have been using is the UK Biobank, which kind of makes sense because it has the largest sample size, the most deeply phenotype data, deeply genotype data in the world. And we use those multimodal data from UK Biobank to answer the question whether the U-shaped pattern between sleep duration and aging clocks can be generalized from the brain to the other body organ system. Like the immune, like the cardiovascular, like the hepatic organ systems. But to validate the U-shaped pattern, especially for the brain, we have used specifically MESA study, which provides the self-reported sleep duration measurements, and also the brain MRI, which like our team has already precised brain MRI imaging data. That's where we kind of validate like the U-shaped pattern between the sleep duration and the brain aging clocks. For research, I think to understand the biology of sleep disorders or sleep disruption is very important. And previously in the field, I think people know, or even the scientists like Dr. Reitland, they know that this U-shaped pattern, which means if you sleep too few or you sleep too long, they are equivalently detrimental for your brain health. And that has been proved in the literature by like focusing on the brain. And here we are trying to extend this concept, try to answer whether this U-shaped pattern can be generalized to a brain body or whole body network. And from our results, indeed, it's not just about the brain, but also like other organ system like immune, like heart, etc., etc. And that's I think the number one takeaway message for me. And the second one, I think from the clinical perspective, we also in the paper we proved that actually the underlying biology or the underlying biological pathway between sleep too short versus sleep too long, they might not totally, but they are very different. For example, we found actually for short sleep patterns, it has been linked to adverse to disease like across the whole body, not just about the brain disorder like sleep apnea. Of course, that kind of makes sense, but also like diabetes, obesity, and depressive disorder. All those things, they are all connected and showing like adverse effects from the short sleep patterns. But on the other hand, for the long sleep patterns, we found like the disease association focusing more on the central nervous system. I think if I remember very well, it can be major depressive disorder, schizophrenia. So that's one pattern we have observed. It seems like sleep long might not be the upstream cause directly causing those like detrimental or disease burden. Instead, it could be some subclinical factors, or it could be bidirectional causality that actually it's a reflecting point. Actually, your body has already got damage from other biological pathway. And to directly test this, we performed like mediation analysis to link short and long sleep duration with late life depression, the major depressive disorder, to ask whether those aging clocks from different organ systems, why does it mediate this relationship between sleep pattern and late life depression. And we found actually for short sleep, this effect is very direct. So no organ clocks, aging clocks mediate this pathway, but that's not the case actually for long sleep duration. We found the brain and adipose aging clocks, I think they mediate around 30 to 60 variants from long sleep duration to late life depression. So that's also confirmed. Actually, there might exist different biological pathway between these two patterns. Clinically speaking, I think in the future, when patients who have sleep disorder, who have sleep issues, they go to the clinic, I think the doctor should treat them differently for those short sleepers and long sleepers. In general, I think my research interest has been always aging and Alzheimer's disease, and the sleep has been one of I think the most fundamental lifestyle factors that are affecting everything, right? Not just about, of course, it has been linked as a risk factor for dementia, for Alzheimer's disease. But in general, I think it's very important to target those potential modifiable lifestyle factors, and sleep is one of them. Other things could be dietaries, could be exercise. We know if you do all those things in a good manner, I think that's going to promote healthy aging and longevity. And my research specifically has been focusing on two parts. I would say the first one is using advanced computational methods like AI and machine learning to define those individual level biomarkers. For example, in this case, in this paper, we define those aging clocks. But we also have some other work to look at, to study, to investigate disease heterogeneity, like to find those subpopulation, what we call cluster or subtypes in Alzheimer's disease. And each individual, we could, ideally, I want to put each individual in a reference panel, in a reference space where each people we can use AI and machine learning to derive dozens of maybe 10 or 20 dimensional score, or what I call dimensional biomarkers, denoting the risk towards future disease development, how fast your brain looks older or younger than your actual calendar age, etc. So this is the first step of my program to define those AI biomarkers at the individual level. And of course, the next step could be try to understand the biology in a specific context. For example, in this paper, we try to understand what are the genetic variants, what are the biological pathways, what are the molecular factors that might affect the sleep patterns, whether sleep too long or whether sleep too short. So those are the two directions that my lab has been pursuing, defining individualized AI biomarkers and using population genetics to understand those biology underlying those biomarkers. From my personal experience, I think to answer a scientific question, you need to go deeper. And I think that's where the portal can be very useful, because for example, in the sleep domain, it's very important you can get the different data types. In our case, we got the data from different organs, different omics, and I think that's where this is a great initiative to share the research, to share the data, to share the tools, and especially for neurodegeneration, it's not a single disease, in my personal opinion. Even for Alzheimer's disease, a lot of researchers, they argue it's a spectrum of not just a single disease, which I totally agree. I think that's where the deep phenotype data, deep genotype data, expanding the different biological layers to study a specific clinical question. For this could be sleep, this could be Alzheimer's disease, this could be dementia, is super important.